From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on April 17th, 2025. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 88th episode of Matters Microbial. I am truly grateful for every download, listen, and view on YouTube. Thank you all for spreading the good microbial word far and wide. Today's topic is very interesting to me because of how microbes communicate with one another and with other cells. Many folks think that microbes are only concerned about their own growth and responses to the environment, but in fact, there is constant microbial chatter in the form of chemical signals like secondary metabolites. It's not quiet in the microbial world at all. One form of bacterial communication is called quorum sensing. Many microbes carry out functions or behaviors only when in large numbers, and they sense that population increase via a chemical language. Now, that is a subject for another podcast, and I will put links in the show notes. However, take Chromobacterium, which makes a purple pigment called violacine, but only at high population densities. Now, through mutation, let's take away its ability to secrete that population signal molecule. That bacterium is now a reporter because it can still hear that signal and respond to it. It just can't make the signal itself. When we place that reporter bacterium next to various possible emitter bacteria, as seen here, you will see that one bacterium, not pigmented itself, causes the reporter to make the purple pigment. I can even get artistic with reporter and emitters, as seen here. The emitter strain is definitely making a chemical signal that diffuses across the plate, leading to the purple pigment in the reporter, and you can even see the gradient. This is a very simple example of chemical communication in the microbial world. But it turns out again that complex communities of microbes are constantly communicating with one another, or in certain diseases, with the host. Sociomicrobiology is thus central, not just to me, but to the microbial sciences. So I'm grateful to welcome Dr. Reed Stubbendieck of the Department of Microbiology and Molecular Genetics at Oklahoma State University to the podcast to discuss the microbial chatter in the cystic fibrosis lung and what it tells us about sociomicrobiology and perhaps how to leverage that conversation into better treatment options. Welcome to the Quality Quorum, Reed. It's so great to meet you.
Thanks for having me.
I think a good way to start for listeners and viewers is your model system. And even though I'm trained as an environmental microbiologist and a microbial geneticist, would you mind talking to us a little bit about cystic fibrosis and the cystic fibrosis lung system that you're looking at?
Sure, I'd be happy to. So this is one of the major systems that we're working on in my laboratory. And cystic fibrosis, if you're not familiar with, is this genetic disease which is caused by kind of a suite of different mutations in a gene known as the cystic fibrosis transmembrane conductance regulator. And these mutations impair the function of chloride ion transport across different epithelial cells. And this affects sort of mucus secretion in many different organs in the body, particularly in the airways. And what happens in the case of this disease is there's this accumulation of a thick and sticky mucus in the airways, and the ciliary action of the epithelial cells is no longer able to efficiently remove that mucus. And that mucus then acts as a really kind of fantastic substrate for the growth of microbial communities. So people with healthy airways, they tend to get microbes that might come in through their lower airways through microaspiration. Just as I'm talking to you right now, I'm microaspirating bacteria into my lungs, but I'm going to clear them out pretty effectively. But in the case of people with cystic fibrosis, these microbes can then colonize that mucus and start establishing these polymicrobial communities. And within these communities, these kind of CF-associated microbes are interacting with each other. One idea that's what's occurring in the lungs of people with cystic fibrosis is that you have these microbes there, and these microbes are actually interacting with each other. And through their interactions with each other, this is influencing population dynamics and ecology within the airway itself. And it's through these kind of ecological interactions that there are effects on the pathogenesis of this disease.
It's really interesting to hear you put it that way because I've heard the idea that we're just a big tube many times and but the fact of the matter is we are a series of ecological niches within us, all through us, on our surface, inside of us, for microbes to inhabit, live with, cause trouble for, all possible things like any other group of ecosystems.
Yeah, I would agree with that entirely. The microbes that live, say, in the airways of people with cystic fibrosis, they're totally different than the microbes that you would find on the back of your hand or in your gut. Each of these sites is totally unique from each other, and it selects for a different kind of microbial community that lives there.
And may I ask a quick question?
Of course.
Would that community change over time throughout the progression?
Yes. So particularly with any of the microbial communities found within the human microbiome in general, they do change over time, with age or during, if you think about the gut microbiome, it could be with the diet that we consume or with progression of disease. These microbial communities are incredibly dynamic. In the case of these cystic fibrosis airway microbiomes, these kind of start by being seeded with bacteria that come from the oral and respiratory tracts. And this, the kind of unique conditions of the cystic fibrosis airway and that mucus will select then for specific bacterial species to become established down there. But this is not a static kind of community. Over time, these interactions that occur between microbes will change those community dynamics, and you can see this sort of reflected in the major bacteria that are isolated from people with cystic fibrosis over their lifespans. So it tends to, when children with cystic fibrosis are younger, they tend to have much more Staphylococcus aureus in their airways, and as they get older, there's a transition and we see a lot more Pseudomonas aeruginosa, which is sort of thought to be the major pathogen in cystic fibrosis.
But again, it is a community.
It is a community, yes. And this is something that we didn't really appreciate until recently with the advent of these sequencing technologies. So when we started looking at culture-independent approaches and characterizing these microbial communities, what we find is it's not just Staphylococcus aureus or Pseudomonas. There is a whole community of microbes that live in the cystic fibrosis airway. And it's not just bacteria too. There are fungal species such as Aspergillus fumigatus, which are also found in the cystic fibrosis airway.
Incidentally, for listeners and viewers, it's very easy for someone who's I'm going to call them a novice micronaut, someone who has only taken a little bit or cursory knowledge of microbiology, to think that everything in nature can grow on nutrient media. It's simply not true. There are many types of microbes that can't be grown in culture very well. Sometimes they need other microbes to grow. Sometimes they need things that can't be provided in culture media, and sometimes we don't know. So culture-independent work that Dr. Stubbendieck is talking about is a really, really important approach to tell you truly who's there, not simply what you can grow in the lab.
Exactly. And these kinds of approaches, these culture-independent approaches, have really opened our eyes to this sort of hidden diversity of the microbial world, which was there the whole time, just we couldn't see it.
Would you mind talking a little bit about the pathology, the role the bacteria have in the pathology of the disease cystic fibrosis? We've talked a little bit about the genetic basis of it, the production of mucus. So can you give us the not so great news?
Yeah, so the not so great news. What are these bacteria, these pathogenic organisms doing? So the bacteria that colonize the lungs of people with cystic fibrosis, many of them are producing toxins and other effectors that induce a strong inflammatory response in the host. And this host inflammation damages the tissues of the airway and the lung. And this is where the major pathology of the disease arises from. Many of these same bacteria, though, can disseminate from the lungs and cause sepsis or other infections in different body sites. Particularly Pseudomonas aeruginosa and Staphylococcus aureus are well known for doing this.
Another question, Reed, is this one. You've mentioned a little bit, or we've talked a little bit about changes that take place during the development of the disease. That is to say, the microbiome in the initial stages of cystic fibrosis differs from later times in life. How about between individuals? Are there huge differences? Is there always a core component? Are there any indicators like that?
That's a great question. We know that the microbiome within an individual who has cystic fibrosis, that changes over time. Particularly younger individuals tend to have more Staphylococcus aureus, and older individuals have more of the Pseudomonas aeruginosa. As I mentioned, there are other bacteria that are found in the cystic fibrosis lung, and these can also have more variation between individuals. So we find that there are certain trends. There's specific species of bacteria that are more likely to colonize the lungs of individuals with cystic fibrosis, but there is some variation there. Particularly though, what is able to colonize the lungs of people with cystic fibrosis, these are going to be microbes that are well suited to living in this environment. As I mentioned previously, we frequently microaspirate bacteria down into our lungs. So these bacteria are coming largely from our respiratory tract and our oral cavity. It was thought for a long time that these oral bacteria were contaminants from sampling, but more recent work finds that these are established members of the microbial community. For example, there's some work from Ryan Hunter's laboratory who showed that oral anaerobes will ferment the mucin proteins, and that fermentation releases short-chain fatty acids, which can feed Pseudomonas aeruginosa.
Well, it's always an ecology, isn't it?
Always.
I wanted to tell you that someone I went to graduate school with, Dr. Rosemary Redfield, worked on Haemophilus influenza for many years and wrote a review article titled Life in Mucus. So that says it all right there. Now,
That's awesome.
Now I have a question related to this, though, if you don't mind.
Of course.
And it's this. I've often had this idea that you have an established community in your gut and you have things that you ingest and then things that leave, right?
Mm-hmm.
So you have things that are passing through, things that are established there that you've co-evolved with, and you've got things that I'm going to call squatters that take up residence. So you've got this yummy layer of mucus. So here's the question. Are they excluding outsiders after this community develops? Is that microbial community in the cystic fibrosis lung? I'm not talking about how it develops over time, but how it responds to potential new inhabitants of that environment.
That's a great question. And we don't know a lot about that right now. So particularly, it's not the easiest environment for an outside microbe to colonize. As mentioned, the nutrition down there, down in the lungs of people with cystic fibrosis, it's largely going to be those mucin proteins themselves. So really the microbes that can live down there have to either be able to degrade that mucin directly or benefit from some of the byproducts that are made by other microbes degrading it. Now, as we've kind of looked at, it seems that antimicrobial production by these common bacteria found in the cystic fibrosis lung is common itself. So these microbes might be making antibiotics that are also inhibiting other competitors, environmental microbes, from colonizing the same site. But it's something that, to my knowledge, we don't know a ton about right now.
If you'll forgive me saying this, we know for a fact that bacteria can evolve in petri dishes, in being transferred from tube to tube. Why not within the lungs? So I don't want to speak to your work like this, but it may be that the initial infective Pseudomonas is different on in a lot of genetic ways from the established one toward the end of life. And I'm curious, there may be no way to study that, but that would be a really interesting question, don't you think?
Yes, it's a really interesting question. We know that the Pseudomonas aeruginosa that infects people early in their lives is genetically distinct from the isolates that you can get from them later in their lives. So there is a lot of selective pressure that is occurring within the lungs of people with cystic fibrosis, which is selecting for specific lifestyles of Pseudomonas aeruginosa itself. And some of these mutations that accumulate, they help it adapt to living in this environment specifically, and it does affect its interactions with other microbes as well.
Very cool. Perhaps the best thing for you to do is talk about some of the explorations in your laboratory.
Yeah, I'd be happy to tell you a little bit about what we're doing there. So in my laboratory right now, this is looking at the cystic fibrosis lung microbiome is one of our major projects. And we're really interested in particularly asking, how do these microbes that live in the cystic fibrosis lung interact with each other? And particularly, what are the kinds of molecules that they produce to mediate those interactions? So I'm very interested in these secondary metabolites, which are made by bacteria and other microbes to kind of mediate these interspecies interactions with each other. And outside of Pseudomonas aeruginosa, we don't know a lot about what these molecules do. Particularly, this is largely due to the fact that we've been thinking about Pseudomonas aeruginosa in cystic fibrosis for a very long time, and it's only recently that we know that there are other microbes that are commonly down there, and they are interacting with each other all the time. So, in my laboratory, we wanted to start thinking about this from the perspective of a culture approach, actually. So we want to see what can we culture, because sequence-independent approaches, they're great to tell you who's there. It can, metagenomic sequencing can tell us what they might do, but we don't know how the actual organisms work unless we can, hold them in our hands on a Petri plate. So this is one of the projects that we work on in my lab, is to try to actually kind of figure out what are these other bacteria doing. And so we've been working with a collaborator at Oklahoma State University, Dr. Erica Luter, whose laboratory previously collected sputum samples. So that's kind of the respiratory mucus from, that's expectorated, so kind of coughed up by people with cystic fibrosis. And her laboratory previously collected this sputum and cultured it on a large number of different media in the laboratory. And then they froze down these kind of mixed communities and set them in the freezer. And what we've been doing is going back into those then and trying to pull out specific bacteria so that we can ask what happens when we put them together on Petri plates to observe kind of interaction phenotypes.
So that's really cool.
Oh, thank you. And I want to highlight here that this work was only possible due to several really fantastic undergraduate students who joined my laboratory. So these 4 undergraduates named Reagan Decker, Helen Zagul, Alexiva Markham, and Ikram Alderwish. They just went through this heroic effort where they went down into these culture collections and struck thousands of Petri plates to build up a collection now that we have, which has about 1,400 individual isolates in it.
Wonderful.
And now,
I was just going to say, it's important to note that one of the really cool things you can do with microbes in general, almost always, is store them cryogenically so that you can come back and look at them later. And that's such an important concept to get to, because imagine someone spends 20, 30 years on something and they didn't keep anything. Well, that really, no one can build on that, but you can with some of these approaches. And I know you're doing the same thing with your individual isolates so that it becomes a resource for later.
Exactly. And we want this to be a resource that is available for the community as well. It doesn't do us any good to keep these locked away in our freezer forever and have no one have access to them. We have this collection then of these isolates, and we don't know what they do right now. So, it's all nice and well to have, a big collection of bacteria in your laboratory, but we want to see what happens when they interact with each other. And we use a particular assay that a graduate student in my laboratory, Nalodri Bomik, developed for our use called a cross-streak assay. In these assays, we can grow our bacteria on media that mimics the nutrient composition of the cystic fibrosis airway, and we grow it as a central streak on the Petri plate. And one of these bacteria grow by themselves for a few days. And then what we can do is perpendicularly to that central streak, we can put target bacteria on the other sides and then observe what the interactions are between these two organisms. And often what we'll find are things like growth inhibition, where one of these perpendicular streaks is unable to grow close to that original producing colony. And this then, that first microbe is hypothetically making an antibiotic. So these are things that we'll chase after later with sort of natural products chemistry approaches to try to understand what's going on in these interactions. So we see these kinds of effects. We see interactions where there's changes in pigment production, we see changes in biofilm formation, and so on. And so what we're doing with these massive library of isolates is taking an approach where we're screening everything and looking for really interesting phenotypes to follow up in the laboratory. And I want to highlight again that this is all work that's driven primarily by 2 undergraduates in my lab now, Alexiva Markham again, and Sydney Marabi, who have recently joined my lab and have worked on this. Yes?
I want to emphasize to listeners and viewers that it's very easy for undergraduates to think that they don't know enough to make a difference. This is not true. This specific type of experiment is the kind of thing that undergraduates can do, but the most important instrument are the eyes and the brain of the student who said, this doesn't look quite, look at the pigment here, look at the growth inhibition there. And it is part of the entire lab's effort. And you're to be lauded for that. What a wonderful project for them.
Thank you. Thank you. I'll tell you, it's some of the most exciting things to happen is when my students come up to me and say, I got to show you this Petri plate. There's something really weird happening here. And then we'll go and everyone, it's really great. Everyone in the lab will stop what they're doing and gather around and look at these Petri plates. And this has been some of the most fun I've actually had as a faculty member, is watching my students become excited and then, seeing the gears in their heads turn of, well, what's doing that? How can we go and take this from a thing we see on a Petri plate to figuring out why it works?
So if you notice here, I'm going to refer to an image here of the late science fiction writer Isaac Asimov, who says that scientific research doesn't progress by some, and I love this expression, damp Greek emerging from the bath shouting Eureka, but instead someone looking at something and saying, well, that's unusual or odd. And that's exactly what your students are doing.
I love that as well. I'm going to steal that from you.
I'll send you the image.
Okay.
All right, when you're doing the cross-streak technique,
Yeah.
is that kind of only a two-element system, or have you come up with ways to look for interactions with more than two? I mean, things have to start somewhere, don't get me wrong.
Yeah. I could imagine ways you could adapt this to more organisms, but we have largely focused on two right now. Like you said, you got to start somewhere.
And it's very fruitful as well.
Exactly. We've taken all of these organisms together. We've done all these kind of interaction assays, and then we find these phenotypes, and these become the fruit of new projects in the laboratory, and where we start getting to understand potentially some of the fundamental biology that's going on within, for example, the airways of people with cystic fibrosis. So I really like taking things out, looking at Petri plates, and then trying to go dissect these interactions molecularly to understand what is going on back in the original system, in this case, the ecosystem being the lungs of people with cystic fibrosis.
So I seem to first off, I understand that this is based on old papers and all the rest, but it used to be thought that Pseudomonas aeruginosa used DNA to glue itself down, essentially, in the cystic fibrosis lung. So I believe that in early cases, actually using DNase, and this is Cynthia Whitchurch's work, it appears to be helpful early but not late in terms of therapy. And this may be exactly with this evolutionary change that takes place early versus late during the development of the community. But the mucus is mostly glycoproteins, however?
So the mucus itself is mostly, yes, it is mostly glycoproteins. You do have other components in there, like extracellular DNAs and things.
So you're looking at these individual interactions, and they've been a driver to your research program.
Yes. Yeah, so we've been looking at these individual interactions with the idea being that by understanding what these bacteria are doing when we culture them on Petri plates, we might understand some of the kind of community dynamics that are occurring within people that actually have cystic fibrosis. So we can't study the bacteria inside of people's lungs easily, so we use these Petri dish kind of model systems, with the idea being that once we find these interactions, what we like to do then is try to dissect how they work, find the molecules that are underlying those interactions. What is that first bacteria making that's affecting the other organism? And then trying to tie that together with what we know about the outcomes of the disease itself, to try to understand sort of the underlying ecology of these diseases. As an example of this, I'd like to highlight some of the work that a graduate student in my laboratory, Nalaj Ribomik, is working on, which actually stems out of these initial observations made by the undergraduate students in my laboratory, where we started with a bacterium that when we put it in the middle of one of these streaks and we put Pseudomonas aeruginosus perpendicularly to it, what we observed was this really interesting phenotype where the Pseudomonas started to turn green. Now, Pseudomonas makes a number of pigmented compounds, but we've determined that this molecule is a particular secondary metabolite, a non-ribosomal peptide called pyoverdin. And pyoverdin is, if you're familiar with Pseudomonas aeruginosa, one of its major siderophores. So these are molecules that microbes use to acquire iron and other minerals from their external environment. And this is particularly important for a pathogenic organism living inside of people because iron is limited in our bodies. We tend to tie up all of our iron in a little tight box called hemoglobin to prevent microbes from having access to it. So competition for iron becomes a major driving force of interactions that occur between hosts and microbes. And so here we have an example of this bacterium that is inducing production of pyoverdin in Pseudomonas aeruginosa. And we thought this was quite interesting because this pyoverdin is not just a siderophore made by these organisms, it's also a virulence factor. So this is work that's been, you can go back into the literature more than 25 years and you'll start seeing hints of it. But pyoverdin production seems to be correlated with virulence in a number of different model systems. And so what we're thinking here, what might be happening is you have an interaction between two CF sorry, I'm trying not to use the abbreviation CF because I don't like over abbreviating things.
Sure.
What we see here is potential an interaction between two microbes that come from the cystic fibrosis lung, and our hypothesis right now is that they might be competing for iron, which is this limiting resource that's in the cystic fibrosis lung, and through that competition, as a secondary consequence, there is increased virulence to the host.
This is very interesting to me because, and feel free to roll your eyes, I often have students look at the kinds of microbes they find in water bottles, and many of those microbes fluoresce under UV, and I suspect that some of them are making siderophores. Not all siderophores fluoresce, of course, but it's pretty common. And it's important for readers and listeners to understand that if you, the host, can restrict this necessary element, iron, for things like cytochromes, that really gives you a leg up against potential invaders that cause disease. So this battle for iron becomes really important. Now it looks like perhaps the microbes are competing with one another for iron, which is fascinating to me.
Yes. And this is not just occurring in the lungs of people with cystic fibrosis. Competition for iron also occurs among the bacteria that live in our noses themselves. So, when I was a postdoctoral researcher, one of the systems I studied was the upper respiratory tract, particularly the nasal cavity. And we found that bacteria called Corynebacterium propinquium make a siderophore called dihydroxynocardamine. And this siderophore dihydroxynocardamine restricts the availability of iron for other bacteria in the nose, particularly coagulase-negative staphylococci like Staphylococcus epidermidis. And similarly, iron is limited in the nose as well. In most body sites, iron is an incredibly limiting resource. So microbes that want to colonize us have to overcome this hurdle. We were just talking a little bit about these microbial interactions that are occurring in the lungs of people with these microbial interactions potentially occurring in the lungs of people with cystic fibrosis. An environment that we know is iron limited, and there's this potential competition for iron that's occurring. And this competition, we now know, leads to increased production of this pyoverdin molecule, which is in of itself a potential virulence factor. And what we can do and kind of tie this all the way back is we've gone and looked in the clinical literature, and we've found that people who are infected by this first bacterium called a chromobacter, similar to the chromobacter that you introduced in the beginning, but without color, people who have an infection with a chromobacter and a Pseudomonas aeruginosa tend to have worse clinical outcomes than people who have either mono-infection alone. So this, is a connection here that potentially what's going on is these microbes are competing with each other for iron. That leads to a virulence factor, and that's worse outcome for the host. I'd love to tell you exactly what the mechanism of action of that is right now, what the molecules are and everything, but we are still working on that. So stay tuned on that regard.
I think it's really interesting the way that you've dissected this out. When you start with a simple interaction between two of these bacteria, you see this reaction where this one compound is being made where it wouldn't be made in quite that large of an amount. And then you look at that compound and decide, and find, I should say, in the clinical literature how important it is. And it's very much, this is the way science is. You find a thread and you pull it and you see where we go with it.
Exactly. Yeah, this has been one of my kind of favorite parts of working in these microbial kind of ecosystems, is being able to look at interactions and how their effects manifest through multiple scales. I think about this a lot sort of at the beginning stages when we were just starting and thinking about microbiomes. A lot of what we were doing was this important kind of natural history work of cataloging which microbes were there and different body sites, there are abundances and so on. But that we can only learn so much by sequencing and asking who's there. We've got to go then take them into the laboratory and figure out why are these distributions occurring? What are, what's underlying these interactions? What is responsible for making these big patterns that we can see? And then, what is the actual, what is the effect of these different patterns at all? And so this is something I've really tried to keep in the work in my laboratory, is being able to start thinking about kind of big scale problems and zoom down to individual molecules and try to understand then what are those molecules doing and how do they have these ripple effects through increasing layers of complexity and scale.
All right, next steps.
Next steps. This is where we go from microbiology to chemistry, which is another major thing we work on in my laboratory, is looking at the secondary metabolites which are made by these bacteria. So this is where, all of the students I recruit, this is where they get a little bit out of their comfort zones. And I tell them that from the beginning that, this is going to happen. We're going to figure it out, though. We got resources. We're going to find these molecules. And I tell them it was actually the same thing that happened to me when I was a graduate student with Paul Strait at Texas A&M. I came into his lab as a genetics PhD student, and he immediately put me on a project that involved a lot of natural product chemistry. So, if I can do it, they can do it too.
Yeah.
So what we're going to do, what we're working on, we're growing up these bacteria right now. We're extracting the compounds they're making. We're purifying them using chromatography techniques. We'll test them to make sure we've got the actual molecules we're looking for. And then that's where we go and we'll use techniques like high resolution mass spectrometry or nuclear magnetic resonance imaging, so mass spec and NMR for short, which we can use to dissect what these molecules actually look like. And from here then, the hope would be that if we know what these molecules look like, we can go back into the genomes of our chromobacter, find the genes that are responsible for making those secondary metabolites, and investigate them genetically. So I would love to be able to, or to delete these genes and see this interaction phenotype then go away, as an example. And as a great experiment. I love genetics for the fact that you can do some of the cleanest experiments in the world. Take a gene, delete it, see an effect, put it back in, restore the phenotype. I love a genetic experiment.
It's so reassuring.
Exactly. Nothing is quite as exciting as when you open up that incubator and you've got your wild type, so your non-mutated strain, your mutated strain, and then the one where you've reinserted your original gene and everything looks exactly like you want it to be. But sometimes it doesn't.
So we've had other people on the podcast who've done what they're calling metaproteomics. That is to say, looking at the whole constellation of proteins that you might see in a given situation. But what you're doing is different than that because you're trying to focus in on some of the secondary metabolites and figuring out what they are specifically, and then learning about what genes encode the enzymes that make those secondary products.
Exactly. This is, yeah. And this is, something that I've always been interested in is these secondary metabolites themselves. What are the genes that are responsible for them? And then how do those genes and how do those molecules influence interactions? And I'll say, I came to this cystic fibrosis project actually going about it from starting at the perspective of the biosynthetic gene clusters that are responsible for making these secondary metabolites. So I'd read the literature and I'd become kind of interested in thinking about this as a really unique ecosystem within the human microbiome. But as I said, a lot of the work had just been done on Pseudomonas and aeruginosa and Staphylococcus aureus. And I was curious, what about the other bacteria that are there? And what we initially did was a genome mining approach. So looking at all the genomes for the common bacteria that were reported to be found in the lungs of people with cystic fibrosis, and then mining their genomes to find these gene clusters. And when we did this, what emerged was that many of these organisms encode biosynthetic gene clusters for secondary metabolites, and we had no idea what most of these do. And so this became really exciting to me to see that there's this huge potential for these interactions that might be occurring, and they might be using these secondary metabolites for it. And it's all in the lungs of people with cystic fibrosis. So that was our initial impetus to do this. Was we had genomes first, and then we wanted to see, well, what are the strains with these genomes doing?
And you've come up with some likely prospects?
Well, we're working on it. This is, where we think about Nalagri's work with this Achromobacter Pseudomonas interaction. That's potentially telling us about secondary metabolites that these Achromobacter are making. We can look at their genomes, and they definitely have the genes to make all sorts of different secondary metabolites, most of which we have no idea what they do. And this is kind of leads also into the work that's being kind of championed by the undergraduate researchers in my lab, looking at all these interaction phenotypes. Because if we find interesting phenotypes, then we can go figure out what these organisms are that are inducing changes in targets, sequence their genomes, and start looking at gene clusters again to figure out, what might they encode. We can also, same thing that Nalagri is working on, we can go and extract compounds and see if we can replicate these phenotypes then without having a producer.
So this reminds me a little bit about the work done with Pseudomonas. And I don't know if you recall this, it's ancient history now. But when people were first working with Pseudomonas as a disease-causing organism, a pathogen, sorry Arturo Casadevall doesn't like the term pathogen, and they had a Drosophila and a Caenorhabditis, fruit fly and nematode system. Sounds crazy, but you don't want to go to mice until you learn more. Long story short, they found if they knocked out quorum-sensing genes, they essentially they became much less virulent in the fly and worm systems and also in the mouse system. So what I'm trying to get at is that maybe some of the things that you're finding with these secondary metabolites might indeed give us some kind of handhold on ways to better manage cystic fibrosis as a disease.
Yeah, I think that is, potential there as well. If these molecules are virulence factors like the pyovertin, for example, and we learn how to potentially inhibit them. So sort of the opposite thing that we're seeing right now. But if we see inhibition of these things, these molecules that these other microbes might be producing then could be developed as potential therapeutics then. And thinking about, instead of targeting organisms with sort of traditional antibiotics, targeting with antivirulence factors. for
Yeah.
example.
Yeah.
Yeah, and this would be exactly what you're thinking with, targeting quorum sensing or any other kinds of kind of interaction or communication systems, really, between organisms as an
It's
alternative to antibiotics. Yeah.
the people in the quorum sensing community refer to some of this as quorum quenching. And there are lots of different versions of quorum quenching, and that some of them lend themselves to therapeutics. And so that's what I'm thinking as I'm hearing about your work is once you understand the nature of the signal, you can interfere with that signal. For example, your Achromobacter and Pseudomonas system, where together they seem to be more virulent. If you can inhibit the interaction or the signaling of the interaction, more accurately, that causes that, you might be able to have some kind of therapeutic that would reduce the virulence, which is a really interesting idea.
Yeah, I agree. This is, I think, the new way we need to start thinking about treating some of these infectious diseases is not just using our broad spectrum antibiotics and hoping to kill all of these pathogens. We'll use the term, but thinking instead about targeting, being more specific, targeting specific pathways or even specific microbes versus thinking about our traditional broad spectrum antibiotics, which have off-target effects on all of the other kind of commensal or mutualistic bacteria that are parts of our other microbiomes as well.
No, I think that for listeners and viewers, we all tend to be reductionist and will think one organism causes a disease. And that was very fruitful in the 1800s and even in the 1900s. A lot of good things were done using that approach. But in fact, the idea of groups of organisms together that give rise to effects negative and positive. In other words, disease as an aberration of ecology is, and that's the future, I would think.
I would think so too. There's a lot of that kind of foundational work is happening in the gut microbiome right now, but I think we're also starting to build up the same kinds of evidence in the cystic fibrosis lung microbiome as well. It's interactions and breakdown of communication that is potentially leading to disease. Or, these could be approaches where instead of killing pathogens or potentially pathogenic organisms, sorry, Arturo, I will, I'll use the term.
We all tend to use it, yes.
Yeah. So instead of killing them or killing these things outright, maybe the future of thinking about and treating these kinds of chronic infectious or chronic diseases is keeping these pathogenic organisms below an effective level where they can cause disease. Having almost sort of a policing by other members of the community that they are a part of. Thinking from the perspective of, I'll anthropomorphize a little bit here. I'll try not to, but it's, but thinking from the perspective of other microbes that are part of that cystic fibrosis lung microbiome, if Pseudomonas aeruginosa is killing their host, that's an evolutionary dead end for all of them. They're, stuck in that body. So.
No, I think that's a really good way to look at it. And I remember, have you read Carl Zimmer's, it's now an ancient essay called The Human Lake?
I have not.
I'll send you a link to it and I'll put it in the show notes. I think I already have in a previous episode, but the point is he's talking about how a lot of the science of ecology was predicated on understanding different levels in lakes, depths, I should say, in lakes, and determining what's living there, and how that also relates to the human body. Different body sites have different microbiomes, etc. In fact, leading up to there are some organisms that simply can't grow without other unrelated organisms around them. And he gives an example from the human mouth, which gives me pause every time I brush my teeth. But it's an important thing to remember. We have a tendency to be reductionist. Now, what's interesting here to me is that you started out with, we mentioned that only a small proportion of bacteria in a given environment can grow in the lab. And that sounds like I'm, negative on culture work, and I'm not, because you do have to get a handle on a lot of things. But from what you learned, you'll be able to now look for these gene clusters in other organisms that perhaps can't be cultivated through metagenomics.
Exactly.
And this gives you some ideas.
Exactly. Yes. And this is one of the things that we love to do is look at things in the laboratory like this, and then look at these metagenomic or metatranscriptomic kind of data sets to try to ask, are these patterns that we are looking at in the laboratory reflective of what's actually occurring in a person? And so I'll say we have not done this yet for the cystic fibrosis work, but from some of my postdoctoral work, we were looking at this siderophore molecule dehydroxyinocardamine made by Corynebacterium in the nose. We were able to go and actually mine metatranscriptome data sets from the Human Microbiome Project as a kind of a fantastic public resource, and find that yes, inside of people, the genes necessary to make this molecule are actively being transcribed. So this is, you've hit it exactly. What we want to do is, take what we look at in the lab and kind of expand it outwards to see what's relevant. So,
and what a wonderful project that involves undergraduates as they begin their journey into microbial sciences, as well as your graduate students and potentially postdocs as well. And I think it's a wonderful all-inclusive group. And sometimes people get a little too focused in their work, and that's not happening with you. So I applaud you.
Well, thank you. Yeah, I think I'm my interests are all over the place. It's zeroing in on one thing to focus on is just not how I work.
Well, and I know you have other projects you're interested in approaching as well, and maybe you want to just a few words about some other projects before we close out.
Sure. So I'll highlight one other project because I want to highlight some of the work of another graduate student in my laboratory, because she's not working on the cystic fibrosis project, and I think she deserves some credit too for the fantastic work she's doing. So not only are we doing work in the cystic fibrosis lung, but we're also working with the upper respiratory tract microbiota. And particularly, up the nose. I sometimes like to describe it as digging for microbial gold.
Oh.
But we're working particularly with this organism called Dolichogranulum pigrum. And this is a gram-positive lactic acid bacterium, which is found almost exclusively in the nose of people. A little bit have been found actually in dog noses and some non-human primates, but this is an organism that we know shockingly little about. Yet you can go back over 15 years to microbiome sequencing studies, and this bacterium is consistently associated with healthy people. So in kids that lack D. pigrum, they tend to develop more ear infections. They're more likely to develop asthma or other wheezing diseases. Adults that don't have high levels of Dolichogranulum pigrum tend to get chronic rhinosinusitis. Chronic inflammation. This bacterium, its abundance is inversely related to Staphylococcus aureus and Streptococcus pneumoniae. This is just an organism that is consistently found to be in the noses of healthy people and less so in the noses of sick people. And we really don't know a lot of why that is. Madeline Reichert, who's a graduate student in my lab, actually a former undergraduate who stayed on in my laboratory as a PhD student, has decided she wants to try to help figure that problem out. So I'll highlight, we are not the only people working on this right now, but we're trying to look at it from sort of a natural products chemistry approach. When we can look at the genomes, we find that they have these secondary metabolite gene clusters. And so we're trying to see maybe this organism is making antimicrobial metabolites that we could potentially develop as new therapeutics, or maybe even think about using and developing these strains as nasal probiotics.
Oh, What a wonderful project.
Thank you.
Now, I don't know if you want to talk about this at all, but I'm wondering if you might say a few words about your path to the microbial sciences. You already described kind of strolling into a postdoc lab as a geneticist and being told to do natural chemistry. We all have that scientific whiplash happen to us at some point. But what was your path briefly?
Yeah. When, reflecting on this question, there's sort of, I think, two moments that I can think about particularly that really made this click. And the first was when I was in middle school. And I remember we had our kind of a common area where all the classrooms are around. I remember my 7th grade science teacher laid out this kind of ring of tape to represent the size of a bacterial cell, and then had a little ball and said, "This is a virus. This is the size of it." And I've never been blown away by the scale. I think reflecting on it now, the scale was probably a little off in that, but it really made me start thinking about these small things. And I was, for a long time after that, really I was kind of obsessed with the T4 bacteriophages, that kind of charismatic lunar lander approach I thought was the coolest thing when I was like, you've got one right there.
I've got lots.
That's great. So that I think was the first thing. And then sort of as the years went on, interest kind of waned in things. And eventually, when I was an undergraduate, I thought for a while that I wanted to be a physician. And then I happened to see this flyer in the building that I worked in where someone needed a research assistant to work on spiders. And I said, you know what? I'll work on some spiders. Why not? So I joined this lab as an undergraduate researcher myself and worked in this kind of the behavioral ecology of wolf spiders for several years. And this got me interested again in thinking about doing graduate school instead of, going to medical school. I became really interested in the idea that someone had to figure out all of these facts and things that everyone learns. And I was like, I want to be that person. I want to figure things out. But I had thought then again I was going to be a geneticist. So when I went to grad school, I interviewed. I was sure that I was going to join a particular lab that was studying Drosophila melanogaster, or fruit fly genetics. And then serendipitously, on the last day, on my exit interview, I met my future PhD advisor, Paul Straight. And, we just got to chatting, it was sort of those like administrative formalities. And then I said, well, what do you do? And then he described the work he was doing, studying microbial interactions between soil bacteria, Streptomyces and Bacillus subtilis. And I thought it was the coolest thing ever, the way he described it, the approaches they were using, the communication. And when I left the interview, got on my plane, it was all I could think about for days and days. And this is just, I couldn't get it out of my head. I was telling all my friends about it. I'm like, this is the coolest stuff I've ever heard of. And so when I started graduate school, I immediately rotated in his lab, and that's sort of the rest was history from there.
So you heard the one true microbial word is what happened.
Exactly.
Yeah, you heard the call. That's what I call it.
I think that's a really apt way to describe it. It was a call because once I heard it, it was, I couldn't get it out of my head, and I'm here ever since.
And this underscores something I've said many times on this podcast for anyone interested in a career in this field, just in general. You will know the path when you encounter it. You really will. Because it has to be a personal thing when it really speaks to you. I know there's a wonderful book, and we've talked about it on the podcast before, called A Feeling for the Organism, which is the biography of Barbara McClintock. And that expression is how she was able to kind of understand, she felt drawn to understand the ideas behind maize genetics, which is horribly complicated, as you know, being trained as a geneticist. But she heard that call. Same thing for you.
Yeah. I mean, I think that's the best way to describe it. Yeah, it was a call.
A feeling for the microbial.
Yes.
That should be. Reed, this has been a wonderful, wonderful exercise in talking about things. What I particularly like about this whole session we've had is you have a level of it that people that are new to the idea of microbiology can love. You've got a level for it for the more advanced, natural sciences, and you've got a level for someone who says, but what are the health benefits, or what are the human impacts? So I think this hits all the spots that might bring together a lot of different interest in this particular session, and I want to thank you for it.
Oh, thank you. This was a lot of fun.
Yeah, I had a heck of a time, and I want to wish you and your lab and your family the very, very best. And I hope to have you on the podcast again to tell us more.
I would love to come back. Once we've solved some of these questions we've got still. We'll be in touch.
Well, it's been wonderful.
Thank you.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. I love to hear from you. Show notes from today's episode, with wonderful links as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Reed Stubbendieck can be found in the Department of Microbiology and Molecular Genetics at Oklahoma State University in Stillwater, Oklahoma. Many thanks as always to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.